Hello everyone and welcome back again. This is video log 2 and this time we'll talk about the histology of glands. Well, glands are specialized secretory cells. They are specialized secretory cells and they are epithelial in origin. So glands are nothing but the specialized secretory cells and from the origin point of view they are epithelial in origin. The glands can be divided in the unicellular gland and multicellular. Unicellular and multicellular gland. The unicellular glands, the unicellular glands are the one which are single cells and they are distributed among the non-secretory cells. So as I said, they are the single cells and they are distributed among the non-secretory cells. Like the example in this case is goblet cell. Whereas the multicellular glands, on the other hand, these multicellular glands, they are formed by the invagination of epithelium. Now the epithelium will invaginate like this and that will form these multicellular glands. And the proximal part of this invagination forms the duct, whereas the distal part of invagination, it forms the secretory part. So you can see this is the invagination of the epithelium which forms the duct. This is the proximal part which forms the duct. And distal part of this invagination, this diverticulum, here forms the secretory part or the secretory unit. If the gland remains in contact with the surface epithelium, now let's say if this gland, you can see it is in the contact with the surface epithelium and it can pour its secretion on the surface. So this type of gland are exocrine glands. These are exocrine glands which are, you can see this duct system and they are having this secretion which are being poured on the surface. So we call them exocrine glands. On the other hand, if let's say the type of gland which pour their secretion in the blood vessels in which the proximal part of the diverticulum is obliterated. So if this proximal part of the diverticulum is obliterated, so they are not opening on the surface, in that case these glands, they will pour their secretion in the blood vessels, and as we all know they are called as the endocrine glands. So exocrine gland which are having their duct system and they're pouring the secretion on the surface, whereas the endocrine gland in which the proximal part of the diverticulum, it gets obliterated and they pour their secretion into the blood vessel, that is endocrine glands. And then we have the third type, that is paracrine gland. Now in case of the paracrine gland, the paracrine gland are the unicellular, they are the unicellular gland and they secrete the substance that affect the surrounding cell. They secrete substance that affects the surrounding cells. So they does not reach the target organ via bloodstream or something. They affect the cells which are surrounding them. Now, example for this paracrine gland will be like enteroendocrine gland of GIT and pancreas. Enteroendocrine gland of GIT and pancreas. So today in this section, we'll talk about the exocrine glands. We'll talk about the exocrine glands. The classification of exocrine gland they can be classified based on the shape and the branching patterns of their duct. Shape and branching pattern of ducts. This can be one way of classifying the exocrine gland. They can be classified on the basis of mode of release of their product. Can be classified on the basis of mode of release of product, or they can even be classified on the basis of the nature of secretion. So first is based on the shape and the pattern of their duct system. So on the basis of the duct system, they can be classified as a simple or the compound gland. When you say simple gland, that means we are talking about the unbranched duct. When there is no branching in the duct system, these are called as simple gland. And the branched duct will classify it under the compound glands. The one with the branched ducts, they are classified under the compound glands. On the basis of mode of secretion, they can be classified as merocrine, apocrine, and holocrine. Merocrine, apocrine, or holocrine glands, which we will talk about. And on the basis of nature of secretion, exocrine gland can be classified as serous, mucous, or mixed. Serous, mucous, or mixed. So that is about the classification on the different bases. Okay, starting with the simple gland. As we know, simple glands are the one in which the ducts are unbranched. I represented the duct in this with the red color, and you can see there is no branching of the duct. You can appreciate the branching of the secretory part in this, but there is no branching of the duct. So starting with the first one, this is called as the simple tubular gland. The simple tubular gland, and the example of the simple tubular gland is crypt of Libercoon. The next one in here, you can see there is a coiled tubular gland. This is called as coiled tubular. The secretory part is coiled, the duct remains unbranched only, and the example for this is the sweat gland. The third one, where you can see the branching only in the secretory part, this is called as branched tubular. This is branched tubular, and we'll find the branched tubular gland in the fundus, that is in the fundic and pyloric part of stomach. Fundic and pyloric part of stomach. So this is about the tubular gland, that is simple tubular, that coiled tubular, and branched tubular. Coming to this one, this is the alveolar type. This is alveolar gland. The secretory SNS is like an alveolus. So this is alveolar gland. And the example for this will be the mucous gland of urethra. Mucous gland of urethra. And then we have these branched alveolar. These are branched alveolar. You can again see there is no branching of the duct as such. And the example for the branched alveolar will be the cardiac glands. The example will be cardiac glands. And also the meibomian gland. So that is about the classification of simple glands. After simple gland, let's come to the compound glands. Now, as we said, what is compound gland? Compound glands are the one which are having the branch ducts. So again, you can see in this diagram, this red line here represents the ducts, and they are all branched. So that they are all compound glands. So you can classify the compound glands in three types. This one is called as the tubular compound gland. This here is alveolar compound gland. We have a tubule here, and these are like alveolus, so we call them alveolar gland, compound alveolar gland. And this one is having both tubular as well as alveolar, so we call it tubulo-alveolar gland. This is tubulo-alveolar gland. So this is called as the tubular gland, and the example for the tubular gland will be Brunner's gland. The example for the alveolar gland, all these alveolus, that is mammary gland and pancreas. Mammary gland and pancreas. And the example for the tubulo-alveolar gland, one good example here will be the submandibular gland. Submandibular gland is an example of tubulo-alveolar gland. So this is how we classify the compound glands: the tubular, alveolar, and tubulo-alveolar. Now coming to the classification on the basis of mode of secretion. On the basis of mode of secretion. So as we already said, on the basis of secretion, there are three types. One is merocrine gland, apocrine gland, and holocrine. Merocrine, apocrine, and holocrine. In merocrine gland, there is exocytosis of secretory granules. There is exocytosis of secretory granules, as you can see in this animation there. And neither cell membrane nor cytoplasm become the part of secretion. Neither cell membrane nor cytoplasm become the part of secretion. That is the merocrine gland. Whereas in the apocrine secretion, the apical portion of the cell along with the secretory product, it is taken off, it is pinched off. So in this case, the apical portion of this cell along with the secretory product is pinched off. Example for this will be mammary gland. The example will be mammary gland. The lipid contents of the mammary gland is by the apocrine type of secretion, whereas the protein component is by the merocrine method only. So it is only the lipid component of the milk which is via the apocrine type of secretion. Whereas in the holocrine secretion, the whole cell is shed off along with the secretory product. In this case, the entire cell is shed along with secretory products. Along with secretory products. The example that is the sebaceous gland. The example is a sebaceous gland. So as you can see in this animation that the merocrine type of secretion where the exocytosis is of the secretory granule, the apocrine type of secretion where the apical portion of the cell is being pinched off, and the holocrine type of secretion where the whole gland is shed along with its product. So let us now begin with the discussion on the histology of salivary glands. So let us first understand the duct system and then we'll talk about the SNI. So let's start our discussion with the duct system. Now let's say this is the main excretory duct here. This main excretory duct gives off lobar duct. The lobar duct which goes inside the lobe, let's say this is a lobe here. Then this lobar duct will divide into, goes inside, it is called as interlobular duct, and the interlobular duct will go and divide into the striated duct. The interlobular duct will go and divide into the striated duct. The striated ducts are the one which goes inside the lobules and in the lobule these ducts they divide into the intercalated duct. They divide into intercalated duct. So let me give them some numbers. That is, the first one here is the main excretory duct. The main excretory duct. This B here is a lobar duct. The lobar duct goes inside the lobe and we call it interlobular duct. Interlobular duct going inside these lobules. These are lobules here. And we call it striated duct. These are striated ducts. And these striated ducts, they further divide into the intercalated duct. They divide into the intercalated duct. These are intercalated duct. So that is about the duct systems. Main excretory, lobar duct, then interlobular duct, then we have the striated duct and the intercalated duct. So let us understand the histological difference between all these ducts. The main excretory duct or the lobar duct, they will have the stratified cuboidal to stratified columnar type of epithelium. Now let's say if this is a main excretory duct, or you can say it's a lobar duct. The lining epithelium will be stratified cuboidal or stratified columnar. Stratified cuboidal to stratified columnar. That is in case of excretory duct or the lobar duct. On the other hand, the interlobular duct will have simple columnar to pseudo-stratified ciliated columnar type of epithelium. So they have simple columnar to pseudo-stratified ciliated columnar type. So you'll see the different level of nuclei because of the pseudo-stratified nature. So it can be simple columnar to pseudo-stratified columnar epithelium. That is in case of interlobular duct. Next in the line is the striated duct. Now there is a reason we call them striated duct. The reason we call them striated duct because of the basal striation they have. Now let's say this is the basement membrane and that is the epithelium. I'm not drawing the entire epithelium here. Now what you will see special about this epithelium, that nuclei is present towards the apex and we can appreciate these striations at the base. So you can also appreciate these striations under the microscope and we call them striated duct. Now why these striations are present here? If you look at one cell here, let's say this is one cell here, as you said nucleus is in here. So there are the foldings of the plasma membrane present at the base. These are the foldings of the plasma membrane present at the base, and between these folding we have mitochondria arranged. Which gives it a striated appearance, and that's why they are called as the striated duct. And this is a typical characteristic of the cells which are engaged in water and the salt transport. So after the striated duct, coming to the intercalated duct, they have short cuboidal cells in the intercalated duct. They are having the short cuboidal cells. You know the property of cuboidal cells will have the centrally placed round nucleus. There is short cuboidal epithelium. And they may be surrounded by myoepithelial cells. We'll talk about the myoepithelial cells later in the section. They may be surrounded by myoepithelial cells. So that is about the histological difference between the different type of duct system you will see in the gland. So you'll see the stratified cuboidal to the stratified columnar type of epithelium which is present in the main excretory duct or lobar duct. The interlobular duct will show the simple columnar to pseudostratified columnar type of epithelium. Striated duct, very peculiar striated duct because of the striations which are present um, in the base of the cell, that is because of the plasma membrane folding, having mitochondria arranged in between. And then we have this intercalated duct, which is having this short cuboidal type of epithelium. Regarding these duct system, it is important to note that striated duct and intercalated duct, striated and intercalated duct are predominantly present in serous gland. Are predominantly present in serous gland. Whereas intercalated duct are poorly developed, they are poorly developed, and striated ducts are absent in mucous gland. Striated ducts are absent in mucous gland. So this is an important point here, that striated and intercalated duct, you will find them, they are predominantly present in the serous gland. They are predominantly present in serous gland. Whereas the intercalated duct are poorly developed, and more importantly, striated ducts are absolutely absent in mucous gland. So after the duct system, let's talk about the SNI. And to begin with, Cirrus SN. Now, Cirrus SN are pyramidal in shape, and they have a round nuclei. They have this round nuclei which is located near the base. Notably, there is a large amount of rough endoplasmic reticulum and free ribosomes which are present close to the base. So this is important to note that there is large amount of rough endoplasmic reticulum and the free ribosome which is located near the base, and therefore they are stained with hematoxylin at the base. So that is a reason they are stained hematoxylin near the base. So let us say if this is one of the Cirrus SNI. So as we said, we have these pyramidal cells present inside. That is the nucleus. So we have this round nucleus close to the base only. And we also said because of rough endoplasmic reticulum and free ribosome, they will stain hematoxylin close to the base. I'll show it to you in the real-time images. So they will be stained hematoxylin near the base. And also to be noted that close to the apex, because of these zymogen granules, they are stained eosinic. So at the apical part or at the apex, the secretory granules are present. These secretory granules are present. That is nothing but zymogen granules. Zymogen granules, and that is the reason they stained at the apical region, which is eosinophilic. So that is about the Cirrus SN. Now coming to the Mucous SN, similarly coming to the Mucous SN, now firstly that Mucous SN, they are usually tubulo-alveolar. They are usually tubulo-alveolar. We already discussed what is tubulo-alveolar. And they have relatively wider lumen. So relatively their lumen is wide, and the cells which are present inside are either pyramidal cells or could be columnar in shape. So usually tubular-alveolar, relatively wider lumen than the Cirrus SN, and the cells inside are pyramidal or columnar in shape. So let us say this is one Mucous SN here. And there we have these cuboidal cells, cuboidal or columnar. In fact, columnar or pyramidal. And there is the nuclei. Now you can clearly see, unlike Cirrus SN, they have these flat nuclei which is present close to the base. So regarding the nuclei, there is a flattened nuclei near base. Whereas if you look at the apical portion of all these cells, they contain mucus. These are Mucous SN we are talking about. So apical portion of this cell, it contains mucus, and this mucus is stored in the form of mucigenous granules. Mucinogen granules. So they have this flat nuclei close to the base, and the apical portion which contains the mucus in the form of this mucinogen granules. And the thing is, this mucus is lost in the HNE preparation, so apical portion is not stained. And that is another reason the lumen looks relatively more wider. So guys, this is about the Cirrus SN and the Mucous SN. We'll see them in the slides. Now let us also talk about the cells which are present between the basal lamina and acinus, and those are myoepithelial cells. Myoepithelial cells are also called as the basket cells. As I said, this myoepithelial on basket cells, their location is between the epithelium and the basal lamina of acini, basal lamina of acinus, and the proximal part of duct as well. Proximal part of ducts. So let us say this is a acinus here with its duct. So this is how myoepithelial cells you will find them. So these are the cells of the acinus, and then it continues in the duct. So these are the myoepithelial cells, and these myoepithelial cells, they contain actin filament, and hence they are contractile in nature. So they contains actin filament, and hence they are contractile. And that is why these myoepithelial cells, they are responsible for expelling the secretions from the lumen of acinus. Because of the contractile nature, they expel, the function is the expelling the secretions from lumen of acinus. So that is about the myoepithelial cells. And let me also tell you about these staining wonders called as Cirrus demillions. Let's now say this is the acinus which is having these serous as well as the mucous cells. So this is the acinus which is having some serous as well as some mucous cells. So the one with these round nucleus, these are the serous cells, and the one with the basal flattened nuclei are the mucous cells. Now what happens is that, when the tissue slides are prepared, usually by the routine chemical fixatives, that is formaldehyde, then these fixative cause the swelling of the mucinogen granule in the mucus secreting cells. And because of this swelling in the mucinogen granule, this pushes the nucleus of the cell toward the basal lamina. We said they have flat nuclei. The flat nuclei is because of this only, because these mucinogen granule, they push the nuclei toward the base. And not only that, these swollen mucous cells, they also push the pyramidal shaped serous cells toward the periphery of the acinus. So let's say the serous cells will be shifted toward the periphery of the acinus like this. And it appears as if the mucous acinus is having this concentric shaped cap on of the serous cells, which is traditionally described as the serous demillions. So the mucinogen cell pushing these nuclei toward the base, and the nuclei of the mucous SNS, they look flattened and pushed toward the base, as well as pushing the serous cells toward the apex, toward one side of the SNS, and it forms like a concentric cap of the serous SL, serous cells on the mucous SNS, called as serous demilunes. So here is the tabulation of differences between the serous SNS and the mucous SNS. So as you already said that the Cirrus SN, they have usually these triangle to pyramidal type of cells present inside, and the nucleus is rounded nucleus which is close to the base. Whereas in the mucous SNS, we usually have columnar cells. They can be tall columnar cells with a flattened nuclei which is are pushed toward the base because of mucogen granules. Cell boundaries are indistinct in case of the Cirrus SNS, where they are distinguishable in case of mucus. We also discussed that Cirrus SNS, towards their apex, they have these zymogen granules, and they have the mucoid material above the nucleus. And these zymogen granules, they are darkly stained with H and E. And in the mucus, because of the mucoid material, it is lightly stained and appears empty with H and E preparation. We already talked about this part as well. The Cirrus SNS relatively have this thin watery secretion, and obviously, as the name suggests, Mucous SNS, they have thick mucoid secretions. Parotid glands are predominantly serous, and sublingual gland is an example of the mucous type of SNS. We have mucous type of SNS present inside the sublingual gland. So that is a difference between the serous and SNS, serous and the mucous SNS. So let's look into the slide of the parotid gland. This is a HNE prepared slide of parotid gland. A parotid gland is the major salivary gland, and it secretes primarily the serous fluid and a little amount of mucous fluid as well. So let's first begin with the stroma, the connective tissue component of the gland, that is called as stroma. So first let's have a closer look of the capsule of the gland. So capsule is that connective tissue which encapsulate the gland, and this capsule divide the gland into lobes. And because of the presence of the connective tissue, the numerous connective tissues, the lobes are further divided into lobules. So let's say this is one lobule you're looking at. These are the lobules in here, which are divided by these connective tissue septa in between. And these are the smallest functional unit. The lobules are the smallest functional unit. Coming to the parenchyma part, the functional component of the gland, which contains the secretory cells and the ducts, that is the parenchyma. So let's have a closer look of the serous cells first. As we know, serous cells are the pyramidal shaped cells, and all these cells are pyramidal shaped cells here. And we have these free ribosomes and rough endoplasmic reticulum at their base. And because of these free ribosome and rough endoplasmic reticulum, you can see it is basophilic. Close to the base, they are basophilic. You can see the round nucleus of the pyramidal shaped cell. These are the round nucleus. These are all round nucleus here. And the base, look at the basal part of the cell here, that is basophilic. And because of the secretory granules toward the apex, that is eosinophilic. Now all this, the apical portion of all these pyramidal cells, that is eosinophilic because of the secretory granules, that is zymogen granules we have there. How they differ from the mucous cells. So let's have a closer look of the mucous cells. So look at all these mucous cells here. Now you can see the nuclei of all these mucous cells, it is flattened, and because of the mucinogen granule, they are pushed toward the base. So these are flat basal nuclei which is present in the mucous cells, and all this foamy appearance, the cytoplasm look foamy here. the mucus has been extracted. The mucus, as we know, in HNE preparation has been extracted, and that is, as I said, is one of the reason why the lumen of these mucus cells look more wider. So this is about the serous and the mucus type of SNS present inside the parotid gland. As we said, predominantly it is serous. We have a few mucus SNS also. Coming to the duct system and starting with the intralobular duct. We know there are two types and they are found within the lobules. One are intercalated ducts and one are striated duct. So first let's have a look at the intercalated duct. The smallest duct that insert into the gland, these are the intercalated duct here. And what you can see that intercalated duct, they have these the low cuboidal cells and they stain lighter than the SNS. We have these low cuboidal cells which are present inside these ducts. These are intercalated ducts and they do have this low cuboidal type of epithelium present. There is another site for these intercalated duct. Now you can see this is the intercalated duct here, and notice the stain here. The stain is lighter than the SNS. Obviously, it is way lighter than the SNS, and we have these cuboidal cells present at the low cuboidal cells which are forming them. Coming to these very peculiar striated duct, as we know why they are called as striated duct. They're called as striated duct. They have this columnar type of epithelium inside, and you can see these basal striations. If you look carefully, the nucleus is not toward the base; it is more toward the apex, and then we have these basal striations, that is infolding of the plasma membrane, which will hold the mitochondria in between. So, because of these striations at the base, we call them the striated duct, and they are surrounded by a lot of capillaries. You can see they are surrounded by all these capillaries here. After the intralobular duct, let's now come to the interlobular duct. So this is one interlobular duct. You can see this one interlobular duct. And we already discussed that if the interlobular duct, they may have a simple columnar epithelium, or usually they have the stratified columnar or maybe stratified cuboidal. Now here I presume this is a stratified columnar type of epithelium. You can see the different level of the nuclei here, which tells you it is stratified. It is a stratified columnar epithelium. And another feature of the interlobular duct, which unlike striated duct, we saw striated ducts are surrounded by the capillaries, mainly by the capillaries, and that we have SNS all around them. But these interlobular duct, they are supported by the thick layer of connective tissue around. Now look at this thick layer of connective tissue around these interlobular duct. There's another interlobular duct here. So the same feature, the stratified columnar epithelium, and we have this thick layer of connective tissue which is encircling them, having all these blood vessels and the connective tissue around it. Well, this here is the slide for the submandibular gland with HNE preparation only. The submandibular gland is also a major salivary gland, and they also secrete more serous fluid than the mucous fluids. So again, going the same way, starting with the capsule. In the stroma, we have this capsule, which is a connective tissue that encapsulate the gland. That is a connective tissue capsule here. And this capsule sends on many trabeculae inside, or the septa inside. And because of these, the connective tissue septa, the lobes is divided into the lobules. So all these connective tissue septa we can see here, they are dividing the gland into lobules. In the parenchyma part, the functional component of the gland, which again we talked about, is containing the secretory cells and the ducts. Now again, having the closer look of the serous cells in these submandibular gland. These are the serous cells, we can see that. The same feature, they are more basophilic toward the base because of all these rough endoplasmic reticulum and free ribosome. And closer to the apex, they are more eosinophilic because of the zymogen granules. Mucous cells, again, the same feature. Mucous cells having these flat basal nuclei.
Foamy cytoplasm, the lumen looks empty and wider because the mucus has been extracted from it. So these are the Mucous SNs here. And in this slide, let's also have a look at the serous demilunes. Look at the serous demilunes. Now, look at these serous demilunes. What we said, what are the serous demilunes? Now, these are the mucous cells we have inside, and because of the mucinogen granule, the nuclei are pushed toward the base. So we can have these, we can see these flat basal nuclei, and also the serous cells are present as a crycentric cap on them. So look at these serous cells which are present as a crycentric cap. So that is an example of the demilune. This is an example of demilune here. There's another demilune here. So mucus acinus with a serous cap on it, that is serous demilune. Moving on to the duct part, and starting with the intralobular duct. Again, the two types we know, that the intercalated duct and the striated duct. So let's have a look at one intercalated duct. You can see this is one intercalated duct which is having this low cuboidal epithelium, and if you look at the staining, it's stained way lighter than the acinus which are present around them. So that is an example of the intercalated duct, that smallest duct which insert into the acinus. Striated duct, we can also see the striated duct here. They arise from the intercalated duct and they have the columnar type of cell, and you can see these nuclei which are not present at the base because of these basal striations. The infolding, we already talked about these basal foldings of the plasma membrane which is holding the mitochondria in between. So these are the striated duct which are present, and again you can see they are surrounded by a lot of capillaries here. Now look at all these capillaries which are present around these striated duct. Whereas interlobular duct, this is the interlobular duct. We also talked about the interlobular duct in the slide of the parotid gland, having the stratified cuboidal to stratified columnar type of epithelium, and you can see this is surrounded by a lot of connective tissues present here. So we have a lot of connective tissue around these ducts, and these are the stratified cuboidal or stratified columnar type of epithelium which is present inside. It's a more closer look to that. So this is a slide of the submandibular gland. And finally, this is the slide of sublingual gland. The sublingual gland is another major salivary gland, and it secretes more mucus than the serous fluid, unlike the parotid and the submandibular gland. So again, starting the same way, going into the stroma. So let's have a look at this capsule. So there is a capsule encircling it. The capsule divides the lobe into the lobules. So these are all lobules this time. These are all lobules, and you can see the connective tissue septa present in between them. In the parenchymal part of the sublingual gland, so first looking at these mucous cells, as we know it is having predominantly mucous cells inside. So all these lightly stained cells with a foamy cytoplasm from which the mucus has been extracted, and that forms the major part of this gland. You can see we have predominantly all these mucous cells present here. This is one mucus acinus you're looking at, and these are those flat basal nuclei. They also have a serous cell. I mean, they are having the serous cells which are relatively darkly stained, and they form the minor portion of the gland. I mean, if you look at the entire slide at once, so you'll find the serous cells are forming the only minor part of this. So these are all serous cells. We already discussed the details of the serous cells, and that's the reason. That's the region where you have this serous and the mucous cells together. So acini which are stained lightly, these are the mucus, and they are surrounded by they're having this cap of these the serous cells. All these serous cap present around the mucous cells. All the muc the the demilunes can appreciate in this region here. So it's a seromucous region where we have these mucous cells which are capped with the serous cells. In the duct system, let me take you directly to the striated duct. Now, even if I'm not able to appreciate the basal infolding of the plasma membrane, I can still say these are striated duct because look at the level of the nuclei. The nuclei is not at the base, so we have this columnar cell, and columnar cell not having the nuclei at the base, that means there is something at the base. And what is that? That is the infolding of the plasma membrane, which is holding the mitochondria in between it. So that is another striated duct. Coming to interlobular duct, the same thing, interlobular duct having the stratified columnar or maybe stratified cuboidal type of epithelium and surrounded by a lot of connective tissue. So there's another example of the interlobular duct. We already saw this interlobular duct in the other slides as well. And let me also show you the main excretory duct of this sublingual gland. Now there is something very special about this main excretory duct. The main excretory duct can have all these, simple columnar or pseudo-stratified columnar or even stratified cuboidal type of epithelium. But that one feature of this is you can see all these goblet cells. Now let me take you closer to it. The goblet cell is a very common feature of the sublingual gland, sublingual uh, s- duct. Look at all these goblet cells present inside the lining epithelium of this main excretory duct. That is the feature of the sublingual duct. So that is about the sublingual salivary gland.